is chapter 15. We're going to be talking about the blood. Let's start with the components of blood. So women and men have approximately the same amount of blood. Men have slightly more. So women 5 liters, men 5.5 liters. If you think about how big a person can be, that kind of is a little bit small. I would probably have guessed that we had more blood in our bodies, but that's it. Women 5 liters, men 5.5 liters. Of that blood, plasma is the biggest component of it. It's 3 liters, and then the rest of it is our formed elements. So the formed elements are the erythrocytes, which we can see here, otherwise known as our red blood cells, our leukocytes, which one can be seen here, otherwise known as our white blood cells, and then we have fragments of cells for clotting, and those are our platelets. So those are all 3 are formed elements. Let's take a look at centrifuged blood. So there's a few terms here. When you centrifuge blood, there's 3 fragments that form. The upper portion is going to be the plasma. Let me just first say that they separate based on weight. So when you spin something, centrifuge something, the heavier substances will fall down to the bottom of the tube, and then they're going to take their positions in the tube according to how much they weigh and how dense they are. So plasma, which is water, is the lightest component of blood, and inside that water, inside the plasma, we're going to have very small metabolites, we're going to have our small proteins, we're going to have our nutrients, we're going to have our hormones. So all of these small little particles or molecules are going to be in the plasma. So this is usually the upper 55%. Then we have a very small layer called the buffy coat. And the buffy coat is going to be a yellowish color. And this is going to be filled with leukocytes. So this is our white blood cells and our platelets. So they're going to be in this intermediate zone. And then this is going to be just about 1%. And then the rest of our blood, the erythrocytes, are about the lower 45%. And these are just approximate, right? So the hematocrit is only red blood cells. And you can take that hematocrit number and determine whether a person is producing adequate amounts of red blood cells. So the reason why your red blood cells are so heavy is because they have iron inside of them, right? So there's that protein called hemoglobin, and iron is part of hemoglobin. So iron is very heavy. So the normal range of hematocrit would be seen here. So for women, something around 37 to 47, and something for men, something around 42 to 52. Now, if you take someone's blood and you spin it, and you find that the hematocrit is low, that means they're not making enough red blood cells. So that's called anemia. There's a depressed hematocrit percentage. And if you look at the opposite, if someone's producing way too many red blood cells, that's called polycythemia. It's an elevated hematocrit. Polycythemia could be normal. Sometimes if you have gone into a higher altitude, or if you live in a higher altitude where the air is quote unquote thinner, there's just not as much oxygen concentration there, your body will actually naturally put out more red blood cells so that the amount of oxygen that's in your body is the same as whether you are in a high oxygen altitude. So these are some variations of hematocrits for you. And just staying on this picture here, let's just talk about plasma. So you should know from your outline that there's 3 major proteins found in your plasma. So your plasma is mostly water. You see that 90% of it's water. And then you have proteins and other solutes. So the albumins, in terms of your proteins, the albumins, globulins, and fibrinogens are your most prevalent proteins. So out of your proteins here, right, let's just put them in this. So the albumins, sorry, I misspelled it, albumin. It is the prominent protein that is going to be responsible for osmotic pressure. Osmotic pressure. We looked at that in the blood vessels unit. And it is also going to be, the other thing you want to know about albumin is that it's produced by the liver. and that was also mentioned in the previous chapter. The globulins, I once had a student write goblin for globulin, which is really cute, but it's globulin. They're globular, so that means that they're roundish. And this is going to be a whole different kinds of proteins have this globular shape. So if you look at your outline, they can be clotting proteins, they are antibodies, they are binding proteins. Remember, we had some binding proteins that held on to lipid-soluble hormones or metals. So globulins are about 36% in your notes. And then we have another really important fibrinogens. I can't write with this pen, but fibrinogens are going to be clotting factors, and they're going to clot blood or coagulate blood. Really important, and we're going to look at that the last part of this lecture. Electrolytes are also found in your plasma, so we can put electrolytes here. Okay, and electrolytes are just charged ions, so sodium, salt, sodium, and chloride, and hydrogen, bicarbonate, potassium, calcium. Those are all in the blood. The most prevalent will be salt. Now, serum. Serum is often used clinically, and serum is basically the plasma. You take someone's plasma and you take out the clotting factors, so minus your clotting factors. Why? Because, so this is serum. Serum can actually contain substances that are used for therapy. So now that we have this coronavirus circulating, people who have gotten infected with coronavirus will actually produce antibodies, right? So they'll have little antibodies are in the shape of the letter Y. So these antibodies, which are a protein, are found in the serum, and they can take a person who had been infected with coronavirus. That person has made antibodies to attack the coronavirus, and then you can give someone else that person's serum. And so you're basically donating antibodies that had been formed in one person, give those to another person who is sick with coronavirus, and those antibodies will fight the coronavirus. So it's a kind of therapy. Clotting factors are removed because you don't want the proteins to clot before you use it for therapy. So it's just a method to just avoid abnormal clots or clots when it's in the outside the body. Okay, so what I was talking about with the coronavirus is called convalescent serum. And the kind of immunity that you're giving someone else is called passive immunity, because the person who is sick with coronavirus, their immune system is fighting the coronavirus, but it hasn't reached a point where it's winning the coronavirus. If they're very sick, the coronavirus is winning. So it's a way of giving that person some other kind of immunity, and it's passive because the person that's sick, that person themselves did not make those antibodies. So anyways, so that's how it can be used. And the same concept can be used for things like antitoxins or snake bites. So we take the antibodies that are in serum and we give them to somebody who is suffering from a specific kind of infection or toxin. All right, so let's move on to our erythrocytes, our white blood cells. This is just kind of an overview of the components of blood. So we're going to talk about erythrocytes, and then we're going to go on and talk about leukocytes and platelets. But this is a good overview of the quantity and what they do. So I like to just focus on function and the anatomical features. You did a lot of this in anatomy, so I'm not going to have you guys recognize these white blood cells because you did that already. So we're just going to focus on function because this is physiology. So if we're looking at the shape of erythrocytes, these are biconcave, right? So they're dented in on both sides. So if we had one on its side, right, we would have a flat, it would be dented in on both sides. So it's kind of like this shape. So it's biconcave. And that shape actually makes it have a larger surface area so that gases can diffuse in and out more easily. It has more surface area. What holds its shape is a very flexible protein called spectrin. And you can see in our little picture here, our spectrin is the red and blue lined up, or the red and blue tangled little proteins. And so there's a scaffolding of spectrin. It fibrous protein cytoskeleton. Red blood cells do not have any organelles. They don't have a nucleus. They don't have any organelles whatsoever. Aside from the cytoskeletal fibers, they are going to be filled with a protein called hemoglobin because their main function is to transport oxygen and carbon dioxide. So I know that a lot of people just say oxygen, and that's 98% true because most of the gas inside of red blood cells is oxygen, but they also carry carbon dioxide. So please remember that it's both oxygen and carbon dioxide. Because erythrocytes can't metabolize, right? They don't have the organelles to basically run the machinery of sustaining life. They are made in the bone marrow. They lose all of their organelles, and when they come out in circulation, they can only exist for about 4 months, about 120 days. So they last about 120 days in your body, and then they're destroyed by your spleen, which is a major organ of destruction, or the liver. And the process of creating red blood cells is called erythropoiesis. So let's take a look at hemoglobin. So hemoglobin, you should know, can transport oxygen and carbon dioxide. So yes, 98.5% of it is binding oxygen, but then it could also bind carbon dioxide. So hemoglobin is a 4 subunit protein. It contains 4 chains of polypeptides. There are 2 alpha chains and 2 beta chains, or you can call them subunits, 2 alpha subunits, 2 beta subunits. Within each subunit, there is a heme group. The heme is an organic molecule, so you can see the organic scaffolding of our heme. But inside, we have an iron. We have an iron atom. So the iron atom is what actually binds the oxygen. So oxygen, right, the molecule of oxygen is held by iron. So iron is a critical component of our hemoglobin. Without iron, it's not going to be able to carry oxygen. So, and we'll look at that in terms of anemia. So it's funny, well, not very funny, but the hemoglobin does change color when it's bound to oxygen or not. So oxygenated blood, when it's bound to oxygen, has a bright red color, as you can see here. And then if it loses oxygen, it's deoxygenated, then it has a darker color. So typically in our artery, in the systemic circuit, the arteries that are carrying our oxygen-rich blood, the arteries will have a brighter red color of blood in them, and then the veins that are bringing the blood back to the heart to get pumped to the lungs to fill with oxygen, our deoxygenated blood will be a darker, so our veins will carry that darker color. Okay, so let's see. We have some of our oxygen is dissolved in the plasma, right? Your notes say 1.5%, but remember that most of it's carried by hemoglobin. All right, what else does hemoglobin bind to? So I said carbon dioxide, but also hydrogen ions and also carbon monoxide. They're all attracted to iron. All right, let's talk about something called carbonic anhydrase. So inside of red blood cells and also in the plasma, there is a molecule, it's an enzyme called carbonic anhydrase. And it's basically, it also carries carbon dioxide, and it's responsible for this chain of chemical equations here, not being able to speak today. But what it can do is it can absorb carbon dioxide. So we can see that here's our carbon dioxide. When your body produces carbon dioxide, right, when your cells make carbon dioxide, it goes out of your cytoplasm. It diffuses right through the plasma membrane into the bloodstream. In the bloodstream, it combines with the water there, and that combination can create carbonic acid. And the carbonic acid is an acidic substance. So in your body, extra carbon dioxide, if you have a buildup of carbon dioxide, that means that you're going to have a buildup of carbonic acid. So too much CO2 will make your blood acidic. So that's kind of down here. In biological systems, carbon dioxide is considered an acid. So this carbonic acid can be converted to bicarbonate. So the bicarbonate, right, is going to be this guy right here. And bicarbonate can free up this hydrogen ion. So what's happening here is that the reason why it's an acid is because you know that acids, just to review, an acid is a substance that donates free hydrogen ions. So the more free hydrogen ions you have, the more acidic your pH becomes. And so this carbonic acid can donate this hydrogen ion into the bloodstream and causing it to be more acidic. Now the opposite is true as well, that if you have a lot of acid in your blood, you can actually drive the equation the other way. So notice how these are all reversible arrows, right? Reversible. So if you have an excess amount of carbon dioxide, you're going to push the equation in this direction, right? Because you have a high concentration of CO2. But the opposite is true. If you have a high concentration of hydrogen ions, you're going to push it this way. So it can balance itself out. So an increase of CO2 can cause acidity in the blood, but then a decrease of CO2 can cause an alkalinity of the blood. So if you're low on CO2, then it's going to drive the equation the opposite way. That's going to deplete your free hydrogen ions, and it's going to lower your pH, which causes an alkaline pH. Okay, so the carbonic anhydrase is going to catalyze the conversion of CO2 and water to carbonic acid. So this carbonic anhydrase is right here. Okay, so it catalyzes the CO2 and water to your carbonic acid. Then that can be converted to bicarbonate. And the more CO2 in your blood, the more free hydrogen ions, the more acidic your blood becomes. Okay, so that's kind of the balance of pH in your blood. Is again, if you increase the amount of CO2 in your bloodstream, it will get pushed, this reaction will get pushed over to the right, and you will start to release those hydrogen ions, causing your blood to be acidic. So carbon dioxide is the same, extra carbon dioxide is the same, it's being considered an acid. Now, if you're low on carbon dioxide, it'll drive the reaction in this way, and that actually causes alkalinity. Okay, so in healthy pH, right, this reaction is balanced, and there's not an excess of CO2, and there's not an excess of hydrogen ions. All right, let's look at the life cycle of erythrocytes. So erythrocyte production in your bone marrow, right? Remember, this is in your red bone marrow. And we have a stem cell called a hematopoietic stem cell, and the process of making blood cells is called hematopoiesis. So this hematopoietic stem cell will diverge into two pathways, and the erythrocytes is going to come from the myeloid pathway. And there are molecules that will stimulate the production of your red blood cells. And so one of the main hormones from your kidneys is called erythropoietin. So erythropoietin from your kidneys is going to stimulate the production of your erythrocytes. And there's different, also other molecules, but just erythropoietin is what is in your notes. And so the developing erythrocytes will produce hemoglobin, and they will eventually lose their nuclei before exiting the bone marrow. So they're going to get, so you can see the pathway, right? They do have a nucleus. They're sort of a functioning, immature erythrocyte, but then right before they leave the bone marrow, they lose their organelles. All right. Requirements for erythrocyte production are iron, because iron is responsible for making hemoglobin. If you don't have iron, you're not going to get hemoglobin. Folic acid and vitamin B12 are both required for DNA replication. So the pathway from the myeloid stem cell all the way to your precursor cells, you have to divide these cells. And every time you divide, you have to copy your DNA. So that's why you need folic acid and B12. Just as an aside, a lot of times folic acid, the other name for it is folate. So if you see folate, it's the same as folic acid. And then vitamin B12 is also called cobalamin. Okay, just in case you see those in the world. Now let's talk about anemia, let's just talk about this one first. So anemia is where your body cannot carry enough oxygen in its bloodstream, and that can be caused by two ways. You can either, one, not have enough hemoglobin, your hemoglobin is low, or the total red blood cell, you just don't have enough red blood cells. So those are two main ways that you can have anemia. Here we have listed just some common forms of anemia. So the most common is going to be dietary anemia. So this is going to be just from an iron deficiency. This one is the most common, iron deficiency. So you're not getting enough iron in your diet because you need iron to, your body can't synthesize iron. It needs to get it from the diet every day. So if you're low on iron, you can be low on the hemoglobin, and you're not going to carry that oxygen around in your blood. Then there's B12. Right, so B12 is required for DNA replication, and your red blood cells are one of those cells that are constantly being replicated. You're constantly destroying millions of red blood cells every day, and you're constantly making red blood cells every day. So you have active mitosis, very, very active cell division. So if you're low on B12, you can also have low red blood cells because you're just not providing the vitamin for DNA replication. Other kinds of anemia is hemolytic. Hemolytic means the red blood cell breaks open. And the red blood cell breaks open sometimes due to an infection like malaria or sickle cell anemia. Aplastic anemia is a bone marrow defect. Renal anemia is from kidney disease, and then hemorrhagic anemia is from bleeding. So you can't... I mean, that's kind of obvious. You don't have enough red blood cells because you're bleeding profusely. All right, so those are just different kinds of anemia. Now let's look at filtering and destroying of our red blood cells. So we can take a look at, I think, this picture. All right, so let's take a look at the step one. So let's take a look at our red blood cell protection, right? So we have erythropoiesis. That's just a more specific way of saying it's hematopoiesis, but more specifically to red blood cells, which is erythropoiesis in the bone marrow, right? Our iron needs to go into our bones so that we can get our red blood cells and our appropriate hemoglobin. So the red blood cells come out into circulation, and they're going to be in circulation for 120 days. And then when they're basically reach the end of their lifespan, they're going to go to one of two organs, the spleen or the liver. So let's start with the spleen. Okay, so the spleen here is going to take in your erythrocytes, and it's going to break open the erythrocytes, and the hemoglobin is going to get processed by breaking away the heme and the globin. So remember, the heme is an organic molecule that contains iron, and the globin is a protein. So globins can be just broken down to amino acids. So those are just chopped down to their building blocks, and then the iron and the heme gets converted to iron, free iron, and then also bilirubin. Okay, so after iron is removed, the heme becomes bilirubin. Bilirubin is actually a yellowish colored molecule, and it's going to travel into the bloodstream to the liver. So we can see bilirubin coming to the liver, and it's going to get processed in the liver. So what happens to bilirubin once it reaches the liver? It can get put into the bile. So that's why your bile is greenish in color, because it's this high concentration of bilirubin. And then some of it will also leave. So you can see from the bile, it comes into the intestines. And then once it's found in the intestines, it is secreted in your feces. Excreted in your feces. So that's the fate of your bilirubin, is that it can get stored in bile, and then it kind of comes out and it's excreted in your feces, but also in your urine. So I don't see a picture for that. Oh yeah, here it is. So bile can come into your kidneys, and it can be excreted in your urine. All right, so what happens to iron? The iron is going to be recycled to make new hemoglobin, and it's transported in the blood, bound to a molecule called transferrin. So you can see the T here attached to iron. That's transferrin. And so it's transported in the blood, and it's picked up by the GI tract or the spleen, and it can go to the bone marrow. So it's stored as a substance called ferritin, which I'm looking for here, but I don't see it. So it's stored as a substance called ferritin. So it becomes ferritin, and that can be stored in your bone marrow, which is where it's used to make more hemoglobin. Okay, so let's see, what did I forget? I also see that my erythropoietin, EPO hormone, comes from the kidneys to stimulate our red blood cell production. All right, last thing here is jaundice. So notice that our bilirubin and the bilirubin is in the bloodstream, bile can be in the bloodstream, but the bilirubin has this yellowish color. And if your liver isn't processing the bilirubin appropriately, it can spill out into the bloodstream in greater amounts. And so it elevates the bilirubin that's in your blood, and since it's yellow, it can cause your skin and the white of your eye to look yellow. So I'll show you the picture here. Okay, so this is jaundice. So jaundice, if a person has jaundice, it's a sign that the liver is having some issues. So very often hepatitis can cause jaundice. Any kind of liver disease or cirrhosis can cause jaundice. Outside of a liver infection, you can actually have a blocked bile duct or excessive breakdown of red blood cells. If you have a lot of red blood cells being broken open, you can have bilirubin buildup in the bloodstream. So this is jaundice. Okay, this picture, I forgot I had this picture. So this is the fate of bilirubin. So we have red blood cell destruction in the spleen. We have the heme protein catabolism, or we have bone marrow erythropoiesis, and those can all create bilirubin. Circulating albumin and bilirubin are unconjugated. That's not super important for us to know, but we can see that bilirubin goes into the liver, right? And then it's secreted from the liver into the intestines, or it can be in the bloodstream to the kidney. So how we get rid of our bilirubin is either through feces or urine. And actually that's why urine is yellow color, because bilirubin isn't found in the urine. All right, I think that's the end of this. So this is, this is another form of anemia. This is misplaced, but this is sickle cell anemia. We're not going into any detail here, but just the fact that the blood cells are unable, because they have a new shape, they're unable to fit through capillaries. And so you're going to limit the capacity of the oxygen being carried to parts of the body if you have a sickling event, and that could cause really severe problems. Okay, so let's review our red blood cells. What is a hematocrit? The answer is letter A, percentage of erythrocytes. Which of the plasma proteins is most abundant and makes a major contribution to the osmotic pressure of the plasma? The answer is albumin. Which plasma proteins include proteins that transport fatty substances and are important in defending the body against foreign substances? This is letter A, globulins. Erythrocytes are the most abundant cell in the blood, but differ from most cells in the body. How are erythrocytes different from other cells? Go ahead and read your choices. And the answer is letter B. What is the function of hemoglobin, the most abundant protein in erythrocytes? Answer is letter C. The development of a particular type of blood cell depends on cytokines called hematopoietic growth factors, which HGF is responsible for erythrocyte production, and where is it made? The answer is letter D. All right, that's the end of part 1.